Hello everyone, my name is Huabo Su from Medical College of Georgia at Augusta University. I'm pleased to be here with Dr. Pan Ma, who is a finalist for the 2025 BCVS Outstanding Early Career Investigator Award. To begin with, Dr. Ma, could you tell us a little bit your background and what initially motivated you to pursue the research at the intersection of cancer immunotherapy and cardiovascular health?
Okay, thank you so much for the introduction, Dr. Su. Hi everyone, my name is Pan Ma. I'm from Washington University in St. Louis. I'm currently an instructor in Dr. Corey Levin's lab. I'm from China, and I got my master's degree from Chinese Academy of Science when I did a tumor adoptive cell therapy. And then I did my PhD in Chinese Academy of Science, where I studied the role of interferon gamma in tumor metastasis and doxorubicin-induced cardiotoxicity. So since then, I got very interested in the immune cancer-related therapy-induced cardiac adverse effects. So I chose to join Dr. Corey Levin's lab as a postdoc, and where I studied how the immune checkpoints affect the heart tissue. Now I'm moving forward to have established my own independent lab in the near future.
Wait, that's fantastic. Your work addresses a timely and clinically relevant question. Could you tell our audience what specific scientific question you're addressing?
As we all know, with the increase in use of immune checkpoints in cancer therapies, so there are more and more patients are at potential risk of the adverse adverse effects caused by immune checkpoints. So my research is to explore the detailed mechanism of the cardiotoxicity that induced by different kinds of immune checkpoints. And hopefully we could translate to clinical to find a biomarker and predictor, or to prevent or predict the cohort that is vulnerable to immune checkpoint inhibitor associated cardiac side effects.
Great. So to address this scientific question, could you tell us how you designed the experiment and what are the major findings that you get from this current project?
Yeah, because in Corey's lab, we have a great platform to employ the novel technologies like a single-cell RNA-seq data, spatial, and the Hyperion image mass cytometry. So I started from the mouse experiment and with these novel technologies, and we hopefully we find some clues that is related to disease progress, and then we validate in the mouse models, and then we back to the patients to hopefully in the future to validate the possibility to treat these mediators, inflammatory mediators in the future clinical research.
So from the presentation you did today, what are the major mediators that leads to the cardiac inflammation when you gave both OX40 activator and also the PD-L1 inhibitor?
The OX40 activation and PD-1 inhibition didn't induce the monocyte recruitment, is not relying on interferon gamma, which is different with the conventional immune checkpoint inhibitors. So what are the exactly mediators? We are still exploring what are the exactly mediators, but one hypothesis is probably CD4 T cells could directly interact with the CCR2 positive macrophages or resin macrophages to make it secrete more chemokines to recruit monocyte and then promote the inflammation in the heart and then increase the myocardial vulnerability.
Great. So from a clinical perspective,
yes.
How can we take advantage of the finding that you make to enhance the patient treatment, especially for those patients that receive cancer therapy and to avoid cardiac toxicity. So could you tell our audience how your knowledge can advance this field?
Yeah, so in my postdoc work in the conventional CTLA-4 and PD-1 inhibitor-associated myocarditis, we found blocking interferon gamma could significantly improve the mouse survival. And also there are also clinical drugs targeting the RUNX, targeting the JAK state interferon gamma signal, and they had a very promising protective effect to those ICM myocarditis patients. So from the OX40 and PD-1 research, my future directions will be identify those pathogenic T cell clones, and I will validate it in the patients by detecting their existence in the PBMCs from the patients, and also compare the common features and also like specific features with the conventional CTLA-4 and PD-1 inhibitor induced myocarditis. This is maybe a good biomarker to predict the vulnerable cohort in clinical.
Wow, that's going to be very impactful. So I just learned that you receive a K99 ROO award, so congratulations on that. So soon you're going to start your own lab, and it seems like you have a long to-do list on your hand.
Yes.
That's great.
Yeah.
Congratulations what you have achieved.
Thank you so much. Thank you so much.